Good morning friends. Today I'll be discussing the topic retina, which I'll be doing in two parts. Now let's look at what are the various topics we have to cover. Firstly, we'll be looking at the structure and composition of the retina, and in this part I'll be also looking at the photoreceptor cells, what is its metabolism, function and its role in phagocytosis. The second part I'll be dealing with retinal neurochemistry, particularly stressing on certain important compounds like monoamines, acetylcholine, GABA, amino acids and taurine and the neuropeptides. I'll be also dealing with the biochemical correlates of retinal diseases. Now let's look at the general concept of vision. When the eye focuses on an image, by projection on the retina, it triggers a series of events. The first event is the photochemical event, where physical events are transformed into chemical or biochemical reactions. These biochemical events then amplify the signals and convert to electrical events. The electrical impulses are then sent to the brain, and the image is reconstructed in the mind's eye, and we see an object. Now let's look at the structure of the retina. The retina is the light-sensitive layer of the nerve tissue. It lines the inner surface of the eye and has been considered to be the innermost tunic of the eyeball. The retina is the one which creates an image which is projected on its surface. It is helped by the cornea and the crystalline lens. And as I told you, first we have the physical events and the biochemical events, and then we have the nerve impulses which are then sent to the brain. It is closely linked with the underlying layers of the eyeball. The thickness of the retina may vary in different sections, and the most important part of the retina is the macula. This ensures central vision and contains numerous photoreceptors known as the rods and the cones, which allows good vision in daylight. Now let's look at the various layers of the retina. So starting from below, we have the retinal pigment epithelium, which is known as the RPE, retinal pigment epithelium, which is known as the RPE. Following that, we have the photoreceptor cell. So please look at the diagram very carefully. First, we have the retinal pigment epithelium or the RPE. The next layer is known as the photoreceptor layer. Following that, the third layer is known as the outer limiting membrane. Next to that, we have the outer nuclear layer. Next to that, as we can see in the diagram, we have the outer plexiform layer. After this, we come to the inner layers. We have the inner nuclear layer, and the seventh is the inner plexiform layer, and then we come to the ganglion cell layer, and then finally we come to the nerve fiber layer, and lastly we have the internal limiting membrane. So these are the 10 layers of the retina which plays a very important role in vision. Now let's look at the first layer, which is the pigment epithelium. Now this extends over the whole optic section. So you can see the retinal pigment epithelium, which is extending over the whole optic section. It borders directly on the underlying choroid. That means next to that you have the choroid layer. Now as you can see in the right diagram, the purple colored retinal pigment epithelium, the shade is made up of hexahedral prisms, and it is arranged in a line. Now this forms a barrier, and this barrier is known as the hematoretinal barrier. Why does it act as a barrier? Because it only ensures certain selective input of substances from the blood capillaries of the choroid into the retina. Now, this retinal pigment epithelium has also phagocytic action. Okay, so the RB cells have phagocytic action. Let's look at the second layer. The second layer is the one which contains the photoreceptors. So it has the light receptor cells. And what are these photoreceptors? They are the rods and cones, and they have been considered the first neurons of the retina. They are the end organs of vision which transforms light energy to nerve impulses. Now, looking at the rod, the structure is regular cylindrical formation and the length is 40 to 50 microns in length. So the rod's regular cylindrical formations and length is 40 to 50 microns, and this rods ensures peripheral vision. The total number is 130 million, and the rods ensure eyesight in poor illumination, like for example night. So it has a high sensitivity to light. The next photoreceptor cell is the cones. They are active in bright illumination. Total number of cones is around 7 million. It ensures central vision and it also ensures color perception. So here we look at the structure of the rods and the cones. Okay. So the rods we can see are elongated narrow cells, and it has 4 regions. The structure, both in the rods and the cones, they have 4 regions. We have the outer segment, the inner segment, then we have the cell bodies and the synaptic terminals. So this is the primary structure of the rods and the cones. The outer segment, the inner segment, then we have the cell bodies and the synaptic terminals. The outer segment in both contain the visual pigments and is responsible for absorption of light and initiation of neuroelectrical impulses. Now the inner segment is the one which contains the cell bodies like mitochondria, nucleus, ribosomes and other organelles for production of energy, synthesis of proteins and other metabolic processes. Now the inner and the outer segments are connected by a stalk of cilium. Then towards the end we have the synaptic terminal, which encapsulates the dendritic tips of bipolar and horizontal cells. So they contain dense structures which are known as dendritic dribbles. The mitochondria is present in the inner segment and in the synaptic terminal. So here we have a comparison of the structure of the rods and the cones. When we look at the rods, on the left hand side is the structure of the rod. We find that in the outer segment, the membrane shelves are lined with rhodopsin or color pigment. So on one side we have cylindrical outer segments and it is longer as compared to the cones. Cones is conical shaped, outer segment, and it is shorter. So in the case of rods, the outer segment is cylindrical. In this case of cones, it is a conical shape and it is shorter in length. In the rods, the visual pigments are arranged as discs. So as I told you, the membrane shelves are lined with rhodopsin or color pigment, and the discs are surrounded by plasma membrane. In the case of cones, the visual pigments are arranged as discs, but the discs are not separated by plasma membrane. Now in the rods, rhodopsin is a G protein coupled receptor and it's a primary pigment. Now in the case of cones, the photoreceptors may be red, blue, green, and it corresponds to or is sensitive to long, middle or short wavelength of light. Now in the rods there is a single synaptic ribbon and slow dark adaptation, whereas in the case of cones, as you can see in the structure, you have many synaptic ribbons and it responds faster to dark adaptation. So this is a comparison, this table gives a comparison between the rods and the cones. Now let's look at the metabolism and function of these photoreceptors. So when we talk about that, what is the main function of these photoreceptors? It is in the visual cycle. Now vitamin A, we have the digestion and absorption of vitamin A. Then it is transported to the retina via the blood. Okay. Now the retinol moves then into the pigment epithelial cells and gets esterified to form retinal esters for storage. When needed, the retinal esters are hydrolyzed and forms isomers like 11-cis retinol, which is then oxidized to 11-cis retinal. The 11-cis retinal will move across the interphotoreceptor matrix to the rod cell, binds to opsin to form the visual pigment rhodopsin. So this diagram gives you the Wall's visual cycle, of which you are very familiar with. In the presence of light, you all know that rhodopsin will break down to give you all trans retinol and opsin. Now this all trans retinal can be converted directly to 11-cis retinal with the help of the enzyme retinal isomerase, which can then bind to opsin to give back rhodopsin. But part of this all trans retinal, what happens to that? It has to get stored. So it then gets converted to all trans retinol with the help of alcohol dehydrogenase in the liver, and it is reduced with the help of the coenzyme NADH, which then gets converted to NAD. But when needed, the all trans retinol can get converted to 11-cis retinol, which then gets oxidized with the help of NAD and the enzyme alcohol dehydrogenase to 11-cis retinal, which then combines with opsin to give rhodopsin. So the visual cycle is one of the main functions of rhodopsin, or the involvement of rhodopsin in vision is through the visual cycle. So when rhodopsin, another thing which you have to remember is, when light falls on rhodopsin or the absorption of photons occur, rhodopsin, as I told you, breaks down to give rise to opsin and all trans retinal. But there is a cascade of events that occur. Rhodopsin does not directly get converted to all trans retinal, but it gets converted to certain intermediates like batho-rhodopsin, lumi-rhodopsin, meta-rhodopsin 1 and 2, and finally it dissociates to form all trans retinal. So this is a cascade of events which occur when light or photon falls on rhodopsin. This cascade of events generates electrical signals, which are then sent to the optic nerve. The nerve impulse generated by the optic nerve is then converted to the brain, conveyed to the brain for integration and interpreted as vision. Once released, the all trans retinal can be converted to all trans retinol, as I told you, and can be transported across the interphotoreceptor matrix to the retinal epithelial cells, completing the visual cycle. Now, how does the transduction of this light signal lead to the generation of nerve impulse, which is then conveyed to the brain? Now, the conversion of light energy to nerve impulses involves three reactions. And these three reactions involves rhodopsin, transducin and phosphodiesterase. And this is known as the phototransduction cascade. Okay, so light energy has to be converted to nerve impulses, and this involves three reactions, and these three reactions involves these compounds, that is rhodopsin, transducin and the enzyme phosphodiesterase, and that is known as the phototransduction cascade. Now, when rhodopsin is photoactivated, it catalyzes the activation of transducin, which is a G protein. Okay, this activated transducin then converts GDP to GTP, and this activates the alpha subunit of transducin. Now, the active subunit of alpha subunit, which is the active subunit of transducin, now dissociates and activates phosphodiesterase. Now, the activated phosphodiesterase will now convert cyclic GMP to 5' GMP. The decrease in cyclic GMP causes light and induces the isomerization of 11-cis retinal to all-trans retinal. This also results in the closure of ion channels in the outer segment, causing hyperpolarization. Okay. So please remember, there are three chemicals which are involved in the conversion of light energy to nerve impulses. The flow of these reactions is known as the phototransduction cascade. That is, one after the other it gets activated and it causes finally the generation of the nerve impulse. So what is first activated? First, once rhodopsin is photoactivated, then it catalyzes the activation of transducin. How does transducin get activated? It gets activated because GDP gets converted. It's a G protein. So GDP gets converted to GTP and activates the alpha subunit of transducin. Now, this activated alpha subunit of transducin will now dissociate and it will activate the enzyme phosphodiesterase. Now, what does this activated phosphodiesterase do? It then converts cyclic GMP to 5' GMP. So what happens? There is a reduction in the levels of cyclic GMP, and this induces the isomerization of 11-cis retinal to all-trans retinal. Also, it results in the conversion of ion channels in the outer segment, causing hyperpolarization. Now, the hyperpolarization which occurs through this cascade of events results in reduced influx of sodium ions. It results in an inhibition of the release of neurotransmitter glutamate at the synapse. It signals the adjacent neurons of the light stimulus. The hyperpolarization is transmitted by the plasma membrane to the synaptic body after phototransduction, and then to the neurons in the retina, and via the optic nerve fibers to the brain. After phototransduction, what happens? The photoactivated visual pigment undergoes decay. The all-trans retinal then gets converted to all-trans retinol. This is transported to the retinal pigment epithelium, and there it is isomerized and oxidized to the 11-cis retinal. This is then recycled to the photoreceptor cells, and the GTP is hydrolyzed to GDP for the next cycle. So these are the various events which occur after the phototransduction cascade has occurred. That is, after hyperpolarization and the involvement of rhodopsin, phosphodiesterase and transducin, you have the formation of 5' GMP from cyclic GMP. So what are the events after that? That is what I have told you in the slide. What happens? How does hyperpolarization finally result in the transmission to the synaptic body, to the neurons in the retina, and then via the optic nerve fibers to the brain? Now this happens when light falls upon rhodopsin. So on the right hand side, we see what happens when light falls on rhodopsin. So you find that there is decreased cyclic GMP, the closure of the sodium channels, the cell gets hyperpolarized, there is a reduced release of glutamate and neurotransmitter, and this is then communicated to the bipolar neurons, and there is hyperpolarization which is occurring there. Now what happens in the dark conditions? In dark condition, what happens? Cyclic GMP will bind to sodium channels in the cell membrane. Okay. So there, in the cell membrane, these channels are then kept open and sodium is allowed to enter continuously, and then for sodium entering there is an efflux of potassium going out, and this keeps the cell in a depolarized state. There is a continuous release of neurotransmitter glutamate at the synapse, and this is communicated to the bipolar neurons. Now what do the cone cells do? The cone cells are used to distinguish colors, and the number of cone cells are less than the rod cells. So the mechanism of stimulation of cone cells, which you can call as red, blue or green, is similar to that of the rod cells. These cone cells are densely packed in the fovea centralis. This is a rod-free area with thin, densely packed cones. There are three types: S, M and L. They are sensitive to short, medium and long wavelengths of light. So each cone has its own photoopsins, which have different response curves. They respond to variations of color in different ways, and that is known as trichromatic vision. So cone cells are specialized for bright and color vision. They are governed by three color sensitive pigments: iodopsin, which is green, synopsin blue, and porphyropsin, which is red. Now what are these? These pigments are complexes of protein and vitamin A. When bright light strikes on the retina, depending on the particular color of the light, one or more of these pigments are bleached, and this then passes the nerve impulse to the brain as a specific color. So the blue absorbs light at 420 nanometers, the green at 535 nanometers, and the red at 565 nanometers. Now the difference in the signals received from the three cone types, that is the blue, green and red, allows the brain to perceive a continuous range of color. So the photoxins may vary in conformation, causing differences in the optimum wavelength that is being absorbed. Now the color yellow is observed when the L cones are stimulated more than the M cones. The red color is observed when the L cones are significantly stimulated. And the blue-violet occurs when the colors are observed when the S cones get stimulated. Now the photoreceptors are metabolically active. They are not dormant, they are metabolically active. And it accounts for much of the energy consumption in the retina. So the retina also requires an energy source. The preferred fuel is glucose, followed by phosphocreatine. It can also use an alternate energy source like lactate, amino acids, high energy phosphate groups. And the major transporter for the glucose is glucose transporter 1. So during illumination, phototransduction and light adaptation, the primary energy which is consumed is in the outer segment. The inner segment has the highest capacity for glycolysis and uses the glycolytic enzymes like hexokinase. It also has provision for oxidative phosphorylation because it has the largest number of mitochondria. It also has a very active sodium-potassium ATPase pump. So the energy which is used, it's used for synthesis, recycling, transport of visual pigments and neurotransmitters. It is used in glutamate synthesis and recycling. And interestingly, more energy is required in the dark, and mainly it relies on oxidative phosphorylation. Thank you. This finishes up part 1.